Very good morning to all. On behalf of the President and Director of BSIP, Dr. Vandana Prasad Ma'am of the Palaeobotanical Society, I, Dr. Pawan will welcome you all to this third Palaeobotanical Society lecture. We thank the speaker of the day, Dr. Arti, for accepting our invitation to deliver today's lecture on the topic Herbarium Techniques and Digitization. The Society aims now to organize lectures regularly and on various topics on the paleobotanical and allied aspects. Today's lecture by Dr. Arti Garg is the third one in the series of the lecture organized this year. And we shall be recording this today's event, and YouTube link for the same will be also shared via email to all the members and staff members. Before we start the main program, I just would like to ask our President and Director, Dr. Vandana Prasad Ma'am, to welcome Dr. Arti Garg and say a few words about the society.
Good morning, everybody. And it's a pleasure to have Dr. Arti Garg for today's lecture. And I hope that her lecture is going to benefit everybody. So I would like to say a few things about this Palaeobotanical Society. As we all know that the Palaeobotanical Society was established in 1946, at the very same year when the Institute, Institute of Paleobotany, was named as BSI Institute of Paleobotany, was also formed the same year. So we can see that both the institute and the society, they were going together, and they were actually the institute was working on research aspects and carrying out research, and the society was disseminating the knowledge of paleobotanical knowledge and other areas also, other allied areas also, and so both botany and geology. So we have seen that it has taken a long way to the society also develop. Institute is also doing very good work, and I hope in near future the society is going to do the work of dissemination of knowledge, and like what we are talking about, like in the previous talks, we have seen a lot of research related issues were discussed. But I also feel that the topics which are for common people, and also the language also can be something like in Hindi also. We can have some lectures which are of interest for many people. So this is what I was thinking, that I think this part we have to encourage more people to become part of this society. So I would request the office bearer, Pawan Gogol, that in fact to send some kind of advertisement, some kind of information to all the departments of the country, both botany and geology, so that we can have more and more people joining this forum. We would also like to have a YouTube channel for the society, and in coming months, what we are planning, that we are going to have some lectures as offline lecture, and that will take place in the auditorium, and the auditorium will be the lectures will be online also. So that we are going to do it, so that we can have interaction with participants also, those who will be attending the lectures, and also the speaker. And so I request everybody to attend these lectures, and also if you can help us in finding out who are the people who can deliver lectures on a more topical areas. And also I would also request the office bearer that if they can have some kind of small interaction or conference or workshop for the students. The students can, they can do everything, and they can find out what are the topics which are very important, and then they can form their. This will be the student will come forward for that. So these are some of the views, and also would like that since it's an online lecture which we are now seeing that many people are liking it, and many people from outside can be linked with us. So we would also request that all of you, if you have any association with people abroad, so we can request them at least 30-minute lecture can be provided by them also. So these are some of my views, and I will not take much time, and let us hear what Arti Garg is going to tell us about on herbarium techniques and digitization. Thank you.
Thank you very much, ma'am, for your kind words and suggestions. We will surely look into it. And just before the talk, I just would like to have brief CV of Dr. Arti Garg. Dr. Arti Garg, Scientist and Head of Office, Botanical Survey of India, Central Regional Center, Allahabad, has made immense contribution in plant systematic and biodiversity. She has completed volume 19 of Flora of India for family, and there are names completed Flora of Chhattisgarh, Thailand, and other ways. And she has significantly, Dr. Garg has discovered 10 taxa, new to science, 2 new records for India, and 1 new record for Northern India, 36 new records from different states of India, and 65 taxonomic names. Dr. Garg has traced lineage of the genus Lodugia and discovered Bacilla calenensis, the axis species from fossil Palaeobotanical records. To her credit are 6 books, 12 book chapters, 156 research paper with a total of around 76 impact factor, as well as several abstracts in conference and deliberations. And now I ask or request rather to Dr. Arti Garg to give their talk.
Thank you, Dr. Pawan. Thank you for that vivid detail about my work. Just one point I would like to stress in this detail itself, that tracing the lineage of Ludwigia and the new species Bacilla calenensis was in collaboration with the BSIP. I think maybe I missed out this line, or maybe so I would like to stress that here. So I would now proceed with the rest of the work and presentation. So at the onset, I would thank your director, Dr. Vandana Prasad, for giving me this opportunity. It's the moment of pleasure as well as pride that I've been a part of this Palaeobotanical Society and a part of the 3rd lecture in the lecture series. So I would now begin with my lecture with a very good morning to all the listeners. Now, I have chosen this topic, Herbarium Techniques and Digitization, for the purpose that it's of common use, firstly, and everybody should know at least the basics of how a herbarium is made, because at a very beginner's level, herbarium looks just a piece of specimen mounted on a sheet, but many of the people are not aware of what is the magnanimous, enormous amount of labor and expertise which is involved in building a herbarium, in bringing the plants from field to lab for future work on which the entire scientific fraternity is based. So from here I would begin my lecture. This is just the basic, that the word herbarium, its origin is very interesting, as finally this word finds its origin from the Sanskrit word bhar. In earlier days, nomadic people used to travel from one place to another in search of green pasture, and during this process they gave the name of green pasture as bhar, which meant to nourish, and slowly from this word bhar was converted to forb was converted to herba. Herba in the real sense means green pasture. And with the evolution of the terms and with the evolution of herbarium itself, the word came into existence in the form of herbarium, which means a collection of book about medicinal plants. So this is where it found its origin. Now, when we talk of herbarium, it's not just a sheet of paper with plant mounted on it. It's a full set of procedure which is involved right from field to lab, as I told. So it's a collection of plant specimens that are dried, preserved and mounted on specimen sheets. These sheets are then arranged through specific system of arrangement, and lifetime they are preserved into herbariums, which may be a book, which may be an almirah, which may be a complete building. So this is what I have tried to show. This is a plant mounted, which is properly dried and then placed in specific folders and brought to almirahs and this entire building of herbarium. So the history goes back to Luca Guini in the year 1490, when Luca Guini, he was the professor in Italy. He was the first person to discover that plants can be dried under pressure, mounted on paper, and then they will provide a lasting record and a complete detail of that plant. So in this book of Luca Guini, there were about 300 specimen herbarium sheets, which now we call specimens, but at that time they were just sheets of papers. And but this this book of 300 herbarium sheets is no longer existing. So at present, the oldest extant herbarium specimen, which was collected by a student of Luca Guini, Gerard Cibo, is existing in the Herbarium of Italy in Florence. And in 1545, the world's first herbarium came into existence in the form of a bound book. This is in the form of a book in the University of Padua, Italy, and this is still existing there. But the process of mounting herbariums and placing them in the book was not found very useful, because for studying one specimen, the entire book had to be extracted, and it was a bit very laborious process. So Linnaeus first started mounting his specimens on single sheets of papers and placing them horizontally instead of binding them in the form of books, so that for study of one specimen, that particular specimen can be taken out, used for studies and placed under a microscope also. Books cannot be placed under the microscope. So this practice continued. It proved very useful, but the process of using books still continued. But with passing time, the usage of books was discontinued and the herbarium started evolving in the form of specimen sheets. So this is the first herbarium building in India. It's Roxburgh House. This building was built in 1793. It is in dilapidated condition. It is still existing in Calcutta. The Herbaria, the Botanical Garden of Calcutta, the building is existing. And later on in 1890, another building was erected, and this building was later modified as the CNH. What we know today is the Central National Herbarium of Kolkata. This is the huge building. It is an L-shaped 5-storied building where herbariums are housed. Now this building, all the 5 stories are having 5 halls. And in each hall, there are about 70 to 80 almirahs and cupboards placed. All these are holding herbarium specimens. You can see them. There are some wooden cupboards, and these are the pigeonhole almirahs which are holding specimen. And these specimens amount to about 3 million. 3 million specimens are housed in Calcutta. Now we come to the types of herbaria. All the recognized herbaria have to have an acronym. Acronym is not a short form. It's a short form given to a herbaria, so that as soon as we say CNH is the Central National Herbarium, but people try to misconceive it for the Herbarium of Central National Herbarium. However, its acronym is CAL. It's not CNH. So this is where some misconceptions have to be cleared. But when we write CAL, all over the world, people know that we are talking of the herbarium, which is located in the Central National Herbarium of Kolkata. And then there are, these are the international repositories. They are very large herbaria, but the national repositories of herbarium are smaller in holding capacity, but still the recognized one, all are having an acronym. So besides these international and national herbaria, there is a type herbarium. The type herbarium houses only the specimens which are called type specimens. Now, this type concept itself is a complete lecture of 1 hour. So I will not go into very much detail, but just for the beginners, I would say type specimen is a specimen to which the identity of any species or taxa is permanently attached. And as long as type specimen is existing, that specimen, that taxa is holding its value. Otherwise, we have to redesignate a type in case it is lost or destroyed. Now, arrangement of herbarium specimens, this I'm just trying to show how they are arranged in Cal. And the arrangement is follows some accepted system of classification. In India, in the Cal Herbaria, we are following the Bentham and Hooker's accepted system of classification. And according to this classification, there are some modifications made in which the geographical region of the particular plant is also designated. So in the arrangement in Calcutta is like, Calcutta means we are talking of the entire BSI, because the entire BSI follows Calcutta pattern. So whenever there is there are 12 geographic zones, and on the basis of these, the herbariums are classified. When we say 1/1, it is India, Pakistan, like that. And then Eastern Asia is given 2 number, 3, like that. We have given 12 numbers to all the 12 phytogeographic zones of the country. And this is one specimen which is showing, here is the GP number 22. GP number 22 is the genera Plantarum. Species number, which genus number, which is placed at 22 number in the genera Plantarum. 1/1 means it is the first phytogeographic zone of India. And here is the generic name with the author, and the number is the family number. Now, when we talk of the important herbaria in the world, the largest one is found, the oldest one exists in France, and it is largest. It has got 100 million, the numbers are in millions, it is 100 millions, then followed by, excuse me, then this is followed by the Kew Herbarium, which has got 80 million specimens, and our country, our Calcutta Herbarium comes 4th in this list. Yeah, it comes 13th in this list with 2.5 million. Actually, the number now is 3 million, but we are holding on record, it is still existing as 2.5 million. The largest herbarium in India again is the Calcutta Herbarium, where in the same series, our herbarium, which is the BSA, falls 10th in this list. We are having at present 1,55,000 herbarium specimens in Calcutta. In Allahabad. Allahabad means this entire jurisdiction, UP, MP and Chhattisgarh, as well as some specimens which may be pertaining to other regions, they are housed separately. So anybody who aspires to consult the herbarium needs to know these basic details, because when they go to the Almira, they are lost. They don't know how to catch hold of their particular species or their specimen which they want to consult. So this is a set of procedures. If we proceed as per the genera Plantarum, then the number of family, we go directly to the number of family, to the phytogeographic zone, and you will directly reach the specimen where you want to, which you want to study and which you want to compare, maybe with your fossil records also. Now, what is the purpose of herbarium? It has got vast use. It serves as reference material for identification help. All the nomenclatural details are written on the herbarium specimen labels. Then it's a comprehensive data bank on diversity and distribution. Here, anything which you find on the label pertaining to its distributional range, its diversity, the characters in field, because some of the characters which are in field tend to get lost when they are mounted on specimens. They get dried up and turn black over time. So all these details are listed on the specimen itself very meticulously, and all details will give you an idea of the complete diversity and distribution of plants over time and over range and in space in the country. These are some of the other usages. In a nutshell, I would like to say that herbaria gives clue to the, if you want to do DNA analysis, you can extract material from herbarium. As we are also collaborating with the BSIP with your director's permission. So many materials are extracted from these specimens, and they are analyzed for future studies, pollen studies, seed studies, leaf, stomata, binding capacity. Gives detail of their distribution and their characters, we cannot proceed. So this in a nutshell is the role of herbaria. Without this, the entire, we can say the entire botanical research can go to a fake output if the correct identity is not there. So from this, we proceed to how these specimens are prepared. This again involves a set of very meticulous procedures. The first and foremost requirement in planning a field tour for collecting herbaria is that a collector should know for what purpose he is going to the field. There can be a general reason, which is called building of a new herbaria, or for writing floras, or for ethnobotanical studies. It is a general collection method which is involved. In the field, the scientists will go, they will make almost all the specimens of all the plants which are found there and make a complete inventory of that region, and they will bring and house it in the herbaria that will build a new herbaria. And yes, hello, should I proceed? Hello?
Yes, ma'am.
Please continue, ma'am.
Sorry, ma'am.
Everything is visible and audible?
Yes.
Okay.
Not a problem.
So that's the general procedure of collection. And if it is a targeted or a specific work, for example, if it's for a study of live plant collection for introduction in gardens, we should know which plant we have to collect, not all plants. If it is a revision, only those plants which are involved in the revisionary studies. If it is an Arti, rare, endangered, threatened plant collection, again, so these are the specific targets. So before going to the field, a set of equipments are required. This I will not go in very detail. These are the set of equipments, including vasculum for plant cutting, GPS, and everything is required. Then blotter papers, absorbent, polythenes, all this is part of the very important point is taking a field book. If a field book is not carried in the field, your entire field trip will go waste, because each field book has got a specific number which is never repeated. Through time, it may be hundreds years, it may be 200 years, but this number is not a repetition. But in that same book, there are 6 sets of these numbers. So if one plant is collected and it is found that this plant is very interesting for future work, we can collect up to 6 replicas of the plant. And each replica is given the same number, which is detached from here and directly tied with the help of thread on the specimen. In case it is not tied on the field, then the number may get mixed up. The specimen may become not useful for future work. So this is, but all these 6 numbers should not be used regularly, because while making collections, we are also supposed to take care of the field where the plants are growing. We cannot keep extracting meticulously all the specimens, but if it is interesting, yes, up to 6 collection can be made. So now I will say what should be collected and how much it should be collected. Now collection again depends on your study object. Whether if you are preparing a flora, then the collection can be exhaustive. Each and every plant samples has to be collected, but it should contain at least a flower, fruit, and vegetative parts. Without these parts, it cannot be called a complete specimen. And for identification, a complete specimen is a must. It cannot be identified unless all reproductive and vegetative parts are there. Sometimes we need to dissect the plant in the field itself, because flowers like impatiens and all, they tend to get shriveled up, and they are not properly, they cannot be dissected properly later on. So for identification purpose, they need to be dissected in the field itself. But very important for taxonomists is that we should not do over collection. We should not damage the diversity. We should not damage the live condition of plants as far as possible. But the population structure should be represented by your specimen. So for representative of, we can collect a representative of the population structure as well as a representative for a species. If it is interesting, again, little more collection can be made. So this I told earlier, at least 6 specimens can be made for special plants. And then these plants are later on dried through a set of procedures, and they have to be mounted on sheets. The standard sheet size is 46 into 42 into 28 centimeter in India. Other countries, some slight variations are there. And then field notes again are very important, because as I told you, in field the plants look different, but on specimen the plant after pressing and drying becomes different in look. So these details should be noted. How much population structure is there? What is the geographic condition of the place, the locality, altitude, the type of plant, color of the flowers, and many more details are to be noted in the field, which will be later on brought to the table. And minute plants like lemna or wolfia can be collected in mass, and then they have to be floated on in water bath. And each and every species is differently segregated, but even the tiniest plant should be mounted on a separate sheet. That is the standard procedure. So these are all the preservatives which are used in defumigation, or which are used in depoisoning of the plant, because when it is brought in the herbarium, it should be completely free of all microbes, of all virus, bacteria infestation, that's all. And for collection of ferns can be collected along with their rhizome, and the hairs and scales of the rhizomes are also important taxonomic characters, so they can be collected. But the large leaves can be mounted in a turned fashion in M, N, or Z shape, or a part of the base and a part of the tip along with the showing the size, how big the leaf is. That is the standard method of collecting ferns. And then similarly for very large plants like bamboo, palms, and banana, there are set of procedures. I may not detail it right now unless it is specifically required. So these are some of the photographs how plants from wetlands are collected. Wetlands are the regions with you all may be knowing about it, where the water depth is about 6 meters, and at frequently this gets infested in water, and all the plants which are found here, some of them pertain to terrestrial habitat, some are strictly hydrophytic, and some are wetlands. So these are almost the transition corridors from terrestrial to aquatic plants, and vice versa. But collection of this is different. We have to use small boats. Big boats will not go because the depth is not very much. So this is the normal dungi boat, we call it. It is used for collecting singara population. But we request the people to take off their singaras and put us in the boat and for collection. So on special request with some money involvement, they will do that. These are some of the field procedures. How the work is to, how collection is done in such dirty places where dungs are also found. So this is just an expert of that. And in field itself, after making few collections, we have to list the complete geographic conditions, the climatic conditions, and details of the plant. This is how we are sitting in the field and making notes there itself. Sometimes when it is drastic regions like Ladakh and high alpine regions, camps have to be placed, and in camp itself lots of work has to be done. And these are the collections. This is my target species of pedicularis collection. Sometimes they grow on rocky areas. So these are just collection excerpts which I have tried to show you. Now, bulky specimen pressing is also a challenge, because fruits like Caparis, Artocarpus, Diosphoros, Balanitis, etc., they require special pressing methods, and they can be sliced, they can be dried after a lot of pressing, after hard pressing for mounting in the sheet. This sometimes the taxonomists have to face challenges like the bale or eagle marbelops fruit and all. This is a really difficult procedure. So this is, I'm trying to show how the pressing is done in the field. This is the condition when a plant is brought to the newspaper or blotters. They should be pressed, and every day they have the blotters or the newspapers in which the drying procedure is followed. That has to be changed every day, and while changing, the plant parts should be properly spread if they are not spread out in the first press. Then over a period of time, say after 7 or 8 changes, the plant slowly gets acclimatized from field to lab, and when abscission layer is not, it's also to be seen that abscission layer may not be formed, because otherwise the flowers and leaves, they will all fall off from the sheet. So this is a set of procedures which is required, and after 8 to 10 changes, it is brought to a field press, nicely tied, and then from there we bring it to the laboratory with all the specimens being tied properly in the field press. So this is just how pressing is done. Nicely, these are tied and hard pressed. Then this is followed by poisoning and preservation of specimens. For poisoning, mercury chloride and ethyl alcohol solution is being used, which is deadly poisonous. A lot of care is needed for depoisoning for poisoning of all these field materials. But without poisoning, we cannot accession them into the herbarium. So this is a set of procedures which is necessarily to be followed. Otherwise, the entire herbarium will get infested with microbes and it will get spoiled. So this is very important step. See how the poisoning is being done. The plant material is dipped in a poisonous liquid for freeing them from infestations. And then the dried and pressed specimens are mounted on herbarium sheets. How we will know that a specimen is dried completely? Because even if the slight moisture is left, it will not, the specimen will not become a permanent specimen. It will not last for longer time. It will get all fungal infection and all will come. So a completely dried specimen is understood when the specimen is lifted from the point end and none of the parts droop down. When the entire herbarium is intact, straight in your hand, then it is understood that it is completely dried. Then again, herbarium label, this is has a definite shape, size. All the details are to be mentioned within, as well as the name of the person who has identified it. This is very important. And then the family, species, genus, species, their author, the nomenclatural details, field notes, everything should be brought on this label. This is the label which carries the complete address and the complete information on the particular specimen. In a summary, I would say that anybody who goes to the field, whether it is a paleobotanist, whether it is taxonomist, whether it is genetic study person, whoever is making a collection, he must keep in mind that a good specimen should always be brought. A good specimen means the specimen which has got vegetative as well as reproductive characters. Without that, we cannot give identification. We cannot give identification certificates also. This is a problem which we are recurringly facing as BSI taxonomists, because from all corners of the country we get specimens for identification, but 85% times it is found that the specimens are not good enough for identification and we have to return them. And these days, even for the thesis, the plant which is used in making a thesis, that has to have an identification certificate which BSI has, which we have to provide mandatorily, but we cannot provide that certificate if the plant itself is not in a good condition, because we ourselves are not sure of identification. So for this, I would request everybody, whoever is, all the research fellows, all the scientists, anybody who wants to take a certificate of identification from BSI, they should bring good specimens so that the identification certificate is also authentic and we can go ahead with that certificate. Now I would detail on the process of identification. Identification at the onset looks like just giving name of the plant. It's not like that. We cannot give name to a plant without going into each and every detail, without matching it with the specimens, without studying the characters in detail and matching it, those characters, tallying those characters with the published character of the species in the form of a protologue. Protologue here is also a complete, means it's a lecture series, so I will not detail much about it. All I would say is the protologue is the first description of the plant. So any plant which has to be authenticated must be matched with its protologue. Slight variation from the protologue would not authenticate it. It may give rise to a variant, subspecies, or it can be anything else but that particular taxa. So from the identification point of view, I would say that it's the rest of the slides will show you how this is followed, and it's a vast procedure. For identification, the first study of characters of the plant is made in the field itself. and an expert taxonomist will be able to come at least to the family level. And coming to the family level means that we are able to now do some visual matching with the plants pertaining to that family. And but this visual matching should not be considered enough for identification. Then after visual matching, after getting the clue of the genus, it has to be, again, with the help of floras and key descriptions, we have to identifying species is the most difficult work. So getting idea of the genus, we have to consult floras and see the entire species key of that genus, follow those characters. Those species key sometimes cut down to the level of the characters of filaments, characters of ovules. So the complete dissection has to be made for identification. Then this of some of the very important point is that the identifier should always put his name on the species, on the specimen field notes, and nothing should be written on the herbarium sheet itself. That has a set of a separate note sheet has to be used for that. Now when we talk of grasses, identifying grasses is the toughest work. Grasses as well as the cyperaceae, sedges, etc. It's a toughest task because, as you see, the characters are so minute. Palea, lemma, gloom, everything has to be dissected out, and properly it has to be studied. It's the characters are sometimes 0.2 mm in size. The Palea, lemma, and gloom keys are based on the size of the palea, size of the lemma, size of the gloom, which varies from 0.2 millimeters to 0.5 millimeters. So you can imagine the magnitude of characters which have to be studied meticulously for identifying grasses, and this is a tough task, I must say. Then after identifying all this, I have in between, I think I forgot to tell about the species cover. All the specimens should be placed in a species cover. Species cover means a cover which has got the, in which all the same species are placed. That is a species cover. Then there, for type specimen, there is a type cover, because every set of specimens, this all I'm trying to tell here is that whatever is there identified, and after identification we have to bring it to the almirahs. In the almirahs, we start from the lowest taxonomic unit to the highest taxonomic unit. So we begin with the species, then genus cover, then family cover, then the cover of the family, and then it has to be brought to the level of Bentham and Hooker system and properly arranged in the cupboards, which are lifetime preserved. Now these are some of the instructions which a taxonomist and everybody has to follow, whether they are making herbarium or they are consulting herbarium. A proper cleanliness is the very root of maintaining a herbarium. Open flames and smoking are strictly prohibited inside the herbaria. Then the almirahs need to be kept closed so that at least rodents and nothing can access inside. Silverfish problem is also very frequently noticed in this, so they have to be poisoned, re-poisoned, again placed back. So this is a set of procedure, a regular maintenance procedure which is followed day by day in all the herbaria of the world, and what we are doing in BSI also. So, and while removing the bundles from almirah, it has to be taken care. Earlier we have noticed some people, when they come to consult herbarium from outside, they are not aware of these procedures. So they simply find their specimen and they drag it out of the bundle. That is wrong. The entire bundle has to be carefully taken out, and each sheet has to be lifted and kept aside. We cannot rub one specimen over the other specimen because that will spoil the plant material. And then pigeonhole almirahs, one pigeonhole should have only as many bundles which can be accommodated inside. So these are for the strictly for the herbarium makers. And another set of cautions is, while putting the bundles back in the pigeonhole, it should be ascertained that the pile of sheet is properly set. They are not falling. The sheets are not falling out and haphazardly arranged in this pile. The specimen should not be kept exposed out of these pigeonholes for a longer period of time, and all the specimens should be horizontally placed. Nothing should be done upside down, and so on. So, and for users, I would say they should never lean with their elbows on the bundle, because that will tend to break all the specimens down the elbow, and there should the herbarium and everything should be kept free of infestations. Now this is one more part of very important herbarium mandates and protocols, that sometimes we as palynologists, seed biologists, and people, they take out the material, and everybody knows, even all the scientists know, that nothing should be done without permission of the proper authority. And here I'm therefore telling you that before detaching or dissecting out any part, it should be also ensured that there is enough material. If a sheet is having just one flower or one bud, we cannot use that for pollen studies, because that bud will, if we take out that bud from the sheet, it will damage the reproductive part. Even taking out one anther, and in extreme cases it becomes very difficult, as I have noticed in studying Daphne, that it just has got four anthers, and if we take out one anther, it becomes deceptive that the flower looks like having three anthers. So these are set of the procedures. So now I will just tell a short summary. From field to permanently preserved sheets involves field collection, drying, pressing, preservation for lifetime. Preparation of type material is has to be specially meticulous. Then identification involves hard work, beginning from having a fair idea through taxonomic experience. Baseline identification, at least to the family level, distributional data, dissections, flora consultation, key consultations, followed by description, matching with herbarium specimens, and writing all detail in the field book. And then only you can get a proper herbarium specimen preserved. Then identification is also done for solving taxonomic complexities in intraspecific taxa. This is very tough. Going to infraspecific level of identification is very tough. And then later on, a revisionary worker can put a determinative or authentication slip on this, which gives the correct identity of the specimen. And if we come to cost calculation of one specimen, a rough idea is each specimen costs around 20,000 rupees, because that involves tool budgets, manpower budgets of scientific team and technical team for preservation, paper materials, herbarium space, which is created for making a herbarium in an institution, day-to-day maintenance, which is a regular work. Every time specimens have to be taken out, dusted, cleaned, put back, sometimes repoisoned, remounting. So a whole set of stuff is employed in day-to-day maintenance of the herbarium, and then air condition has to be given, otherwise the specimens become brittle, and recurring repairs and remounting, preservation and nomenclature updation. So a rough idea with scientific, we cannot come to exact calculation, but roughly it costs about 20,000 for each specimen. And if we talk of the worldwide index of herbaria, there are 3,100 herbaria with associated staff of 12,000 people working for maintaining these herbaria, and the botanical specimens are about 390 million. We have extracted this data from online source. And finally, I would like to say that for any identified herbarium specimen, the identifying person has got at least 20 years of experience, and then only a taxonomist is carved into an identification expert for giving name of that specimen. Now the points to ponder here are that, are herbaria redundant? It sometimes it is also understood in scientific world that what is the need of herbaria, because people who are doing genetic diversity, plant breeding, molecular, they will not understand the value of herbaria. So this, in my ongoing, on my entire lecture, I have tried to show, and I've tried to express that herbaria are not redundant. They are the most important repositories of plant wealth of the country, and without that, the entire scientific work will be baseless. So you can imagine, if the name is not correct, what will be the future of science, what will be the future of the new variety being developed, or any plant breeding or molecular work if the name itself is wrong. If we are working on solanum but we call it lycopersicum, still that is a confusion. Now then, identification is also is regularly understood as just giving the name of a plant, because many times I hear that what taxonomists have done, they have just given name of the plant. So from the back entire presentation, you can understand what is giving name to a plant. Then providing an authentic specimen material. This is also usually conceived as just giving the material. It's not just giving the material. It is bringing the material from field, authentic material, to the lab. So a taxonomist, an expert who has at least 20 years of rigorous experience can give the correct name. Then past versus present, the entire vegetational pattern, if we want to study, the present vegetation is right there in front of us. We can do that, we can study that, but where will the past material come from? Past material comes from herbaria. So this point is again linked. Are herbaria redundant? No. The entire herbarium repository, we have got the oldest herbarium, oldest specimen lodged in our Calcutta Herbarium, the CAL acronym, or the Herbarium of Central National Herbarium, which is dating to back to 1890s. So if we study that specimen of 1890 and we study the same species of today, we will understand the range of variation or the impacts of climatic aberrations or the impact of the entire environment on the plant shifts, etc. They all can be compared only when we have the past data in front of us, and that is what herbarium gives. That is what herbarium gives. And finally, I would like to say that taxonomists should not be underestimated for their contribution. And this is what sometimes the taxonomists have apprehensions, that we are underestimated for our efforts in making a herbarium specimen. So value of authentic specimen is that all research would be fake if the specimen identification is wrong. If the specimen number is not available, then the meaning of the entire science will be fake. The past vegetation cannot be traced without herbarium data, and that's the role of BSI in all plant-based researches and collaborations. So I would just finish my lecture over here. I will not take much of the time because rest is not relevant to the present present work. Now I will come to touch the point of digitization, which is not here in form of presentation, but verbally I will be talking it. BSI now is trying to digitize the entire herbarium, at least those specimens which are called type specimens. They have already been digitized, and they are now available in virtual mode. A complete link is there. Anybody can approach the BSI for obtaining that link. And about 1 lakh specimen have already gone in virtual mode. They can be accessed, and our Prime Minister was very happy. He launched the digital of the herbarium, and he gave a visit to the BSI, and at least more than 1 lakh people till date have accessioned our virtual site. And the process of digitization is a complete set of work which is involving barcoding of all the specimens. They have almost been completed. Barcoding work is completed. All the specimens are being scanned. We started from the type specimen. We have come to the general herbarium, and type specimen are already available online. Anybody who wants to match their specimen or access the type specimen, they can visit the BSI virtual herbarium site and go to the virtual media and get a clue of the specimen. So this is where I would say digitization is now very important. We all are working with the entire team. Every day we are scanning the specimen and uploading them on the virtual site. So digitization has taken good pace in BSI, and very soon we will be having all the specimens of BSI available in virtual mode. Thank you for your patient listening.
Thank you, Dr. Arti ma'am. And now presentation is open for the discussion. If anyone is having any question.
So, Arti, we really enjoyed the talk, and it's really interesting because many people are working and they are taking help from Botanical Survey of India. And so we would like to know if, like, suppose we want a specimen from the from BSI, what are the guidelines and what should we do? How we should move forward? Like how to like a duplicate specimens if we want to like do the paleontological study. So how to move forward?
Yeah, I will expand upon it. Firstly, I think I am not visible to you. Is it true? Because my video, I think, is not. Am I visible to you?
No.
Okay, then I will talk in this just in audible mode. My people are trying to switch on the video mode, but I think there is some technical fault. Anyways, so Dr. Prasad, as you just now wanted detail about this, I would say that procuring specimen from just a moment. Yeah, that's good.
Now you are visible, ma'am.
We can see you.
Thank you to my people here. So I will expand upon this. Whenever we want to do work on, say, pollen seed, any material which is required from the BSI, there is a document in the form of document we would like your request to us. Sometimes if a duplicate specimen is available with us, we will extend it happily from the duplicate specimen. But in most of the cases, because duplicate specimens may not be available, they are not so properly maintained. A collaboration comes into effect where we do in partnership this work. We will provide the authentic material and you can go further with this with authorities. And a third set of procedure is there that it may be in the form, just in the form of acknowledgement, but that is through a complete set of paperwork which involves an MOU where it has to be mentioned that you will not give us authorship, but it will be acknowledged. So in such cases, we may not be able to share very valuable specimens, but the general specimens. But when it is an MOU signed between BSI and BSIP, then we are equally responsible for providing every important specimen that you require, whether we have it as a repository in our herbarium or we have to bring it in from the field, authenticate it, and then extend it to you. So this is it's not difficult. It's just a set of paper movement work.
Okay, thank you. It's a very, very important, I think, information. And I think many of us, we are sitting here listening about listening, so we can like think about that whatever is required, people can move forward. And then it's like what you said that we should have MOU with the BSI. And I think we have a MOU with Herbaria.
Yes, we have done a lot of work under that MOU, like Ludwigia and Basella carolensis. Lot of work we have already done, and it is still in progress. We have already supplied material to the team comprising of Dr. Anjum Faruqi, Dr. Swati. So they are still working on that, and papers are coming out. Very good collaborative research work is coming out from that collaborative work, and we would be happy to enter into the next collaboration also with you all.
Okay. Thank you, Arti.
Thank you, Doctor. Once again, I would like to thank for giving me this opportunity to speak in the series of lectures of Palaeobotanical Society. I am highly privileged, highly honored, and it's really an opportunity to talk about the herbarium with the entire BSIP set of scientists and fraternity. Thank you very much.
Ma'am, there is one more question.
Yes.
Good morning, ma'am. Good afternoon, ma'am. Ma'am, it is really an excellent lecture that I got through, and I just have one curious I need to just know one information. Ma'am, you told about phytogeographic region.
Yes.
So, ma'am, what is the extent? How you define that? I just wanted to know the phytogeographic region for collection.
Yes, that curiosity is yes, it is very justified. The entire country, India, is divided into 14 phytogeographic regions, and there is a set of criteria on the basis of which phytogeographic zone is delimited. That again, it's a complete lecture, I would say here, but in a nutshell, I will tell you that based on the diversity of plants, the extent of spread of rare and threatened species in that particular area, and the climatic conditions which are prevailing in this phytogeographic zone, on the basis of which these plants are determined in this area, which plants will grow, along with it a set of operational threats like landslides, the environmental factors, and species keep on getting lost and another set of species keep coming. So these are the phytogeographic regions are based on the climatic conditions along with the phytogeographic conditions and the set of plants, both general and rare endemic threatened plants which are prevailing in that zone. So if we go from one phytogeographic zone to the other zone, you will not find that set of plants. Like if we talk of Western Ghats, you will not find most of these plants in eastern regions or in the Thar areas of Rajasthan or in Kashmir, Ladakh. So on the basis of this, a set of 14 phytogeographic zones are delimited, and it will take 1 hour to go into the complete detail of those zones. But anything you want to talk to me in person about it, I am free. You can interact finally.
Yes, ma'am, I have one query.
Yes.
Hello?
Yes.
Thank you, ma'am, for such informative lecture. Ma'am, I'd like to ask you that when we are collecting a well-identified plant, then also it is necessary to compare it with its protolog?
Well, this is a very relevant question. If we are talking, I think you are talking of common plants.
Yes,
when it is a common and well designated, well identified plant, it is not necessary to go to the protolog, but it is necessary to go to the descriptive taxonomic description of that plant. Because sometimes, simply if I say Solanum nigrum, you know it's Solanum nigrum. But when you see the description of Solanum nigrum, if one character, say if the filament character or if the ovule setting is different in your plant, then it may be a variant or a subspecies or a vicariant of that plant. So for taxonomist, I would say we will always go to the taxonomic description, because for giving certificate it is very crucial. However, for using it in regular day-to-day works, as taxonomist experts, we need not go. But other field people must be sure of what they are using for their future research.
Actually, ma'am, I asked this question because one of the results when I submitted one paper of pollen morphology, they asked me to compare the specimen with the protolog.
In that situation, the reviewers must have found this species is not strictly matching with the species name that you have given in your paper. So you have to be sure in this situation you have to match it with the protolog.
Because that paper is in collaboration with your organization, BSI.
Okay.
So they have asked this thing that you have to compare the specimen strictly with the protolog.
Okay.
So that's why this is one of the queries which I was finding difficult to answer.
So nice. If it's a collaborative work between BSI and BSIP, then you give us that query. We will match it with the protolog and we will come to a conclusion.
Okay.
Right? Yeah. Anything else?
Is there any other question?
Hello, ma'am. Good afternoon, ma'am.
Good afternoon. Is that Arya?
Yes, ma'am.
Okay, nice to see you.
Ma'am, just one question. Ma'am, I just want to ask that sometimes what we add, a word we add with the specimens, Herbarium specimen, that is paratype or isotype. Ma'am, can you please tell me the difference of these terms? I just forgot that.
Yeah, Dr. Arya. This question, I will say, needs 1 hour of lecture. Because it's based on a complete type concept. So if I get an opportunity in one of my later on talks, I will explain the type concept. But if a holotype is written, I will say that specimen means the protolog on which this species has been named. Holotype means a complete replica of, means it is a specimen which is permanently attached to your description. That is a holotype. Then if we say lectotype, that means the holotype has been lost or destroyed for some reasons, and another set of specimen has been taken into consideration to replace the nomenclatural holotype. When we talk of paratype, paratype is a set of specimen which was studied by the original scientist while describing this species. If he studied 10 specimens, but out of those 10 he made one specimen as a complete holotype. The rest of the specimens which he used but he didn't mention them to be holotype, they will all be paratypes. So if holotype, lectotype, everything is lost, then a paratype can be used to replace the holotype, and it is again called a lectotype or a lectoparatype, and it's a complete set of you say this type concept. There is a one complete book on type concept which is revised every 4 years. There is a conference which is based on making type concepts, and when the type concept there is a complete conference with all the countries participating into it, the type concept may be changed, alters, or retained as such. So this as Dr. Arya, you were once taking some training from BSI, you may be also aware that type concept some lectures were made. So it's a huge work. It's a huge work, and it needs to solve every query. It needs a 1 hour lecture. But broadly, if you have any personal question, you can make a phone call. Ask me. I will tell you what that means. Whether it's paratype, lectotype, isoparatype, isolectotype. Many terms are there which a very trained taxonomist only can explain you.
Yes, ma'am.
Right.
Okay, then I just would like to thank you, ma'am. I'm very thankful to today's speaker, Dr. Arti ma'am, for the informative talk, and literally lots of knowledge being shared. And all the participants, thankful to all the participants to spare their time and graceful presence. And we'll communicate further for upcoming tasks. Once more, we are very thankful to you, ma'am. Thank you.
Thank you. Thank you so much. I may take your leave. Sure.
Yes, please.
Okay.